بسم الله الرحمن الرحيم. Good morning, ladies and gentlemen, fellow speakers, and good morning to our audience joining us online from around the world. First, may I thank each of our speakers for taking part in the event. The quality and quality of speakers this year is outstanding. I also want to thank all of you for connecting the morning, and I hope you are looking forward to the discussions as much as I am. The era of biotechnology and personal medicine is the 10th conference of the Society of Stem Cells and Molecular Biology, in cooperation with the Faculty of the Agriculture Summer University in York. So, Professor Dr. Basim Khamis Kotli is kindly giving a word of welcome. Please, Dr. Basim Khamis Kotli, kindly. دكتور بس المايك دكتور ما واضح. دكتور العفو المايك دكتور باسم بس اصلاح المايك من يمك دكتور العفو. تمام اوكي.
Thanks. It's given me pleasure to welcome from the splendid scientific exec of the knowledge of leadership, International Scientific of Arab Society and Simsal. This began of opening on maybe the blessing, mercy and blessing of God of Abin to after. Proceeding from your device constitution, which is embedded of the verses of the proceeding from the constitution, which is are indicated of the sign of the Almighty. As we assume of the responsibility of supporting of the genius of the conference, we oversees of the first in focusing of equality of scientific research leadership, and preparing of participation leadership to the researcher by developing their skill and ability to learn of earning from the knowledge, to developing the skill and ability of the learn and making of the knowledge to become a national leader in the future. Our conference are aspire to dissemination of the enhanced language and expanding to base of scientific in the library studies and keep aid in the development of developed countries in all fields. Annual of a constitution of the discoveries and invitation and receive of Iraqi humans, civilization and history. So our country need today more than ever in advance of educational and researcher processing and embedded of possibility and Ministry of Higher Education and Scientific Research with any government program in use of his auxiliary of the minister in the beginning of the minister's higher education integration. So, as a basic access in the teaching of learning process and adaptation of the basic concept in the quality of education, such as knowledge acquisition, production of transfer, as well as promoting of the culture, innovation, and creativity. These are the main pillars of the generation of the capable to the working, building, and knowledge in Iraq, and the goal of which is innovation and modernization to the resistance of Iraq. The acceleration steps toward achieving scientific superiority, Iraq University, and adaptation in the scientific research in the international and Arab society and Simsal. So, the basic principle and platform of inter-scientific cross-fertilization in connection to the innovation, science, and technology in parallel to the university and solar conference, courses, workshop, and research within solar scientific impacted through the presence of researcher and their participation in the Iraq of higher education. So, we have found in the scientific primary goal as a result of where international scientific journal a number of patent and the conception fintech in the fantastic of the intercommunication and classification in the international university bodies within principles of university worker. So the Iraqi marker unique invention in the following up of graduate and putting in the container with the adaptation of the project of government program in the science of inception in taking of important steps in the implementation of recording to working access in the mechanism of the adaptation with the vision of the Ministry of Higher Education and Scientific Research in Iraq, as well as the following up in the position and achieving upgrading and requirement with the work of training, twinning and university with the similar of Iraq and international universities, solar of the academic research and institution of exchanges, experience, innovation of scientific in various disability. From the up, there are many structural which is generation to the presence of the benefit to the means of technology and modern scientific provided of university with its making, teaching, recording the student during their studies through the use of various educational means related with technology program, which is given students to the presence of some higher education and scientific research with its duty standard and adaptation of quality, solarity, and providing a property to the lay of the foundation and appropriation. The participation of building educational and scientific effect, which is authority and authenticity with the presence of Iraq to seek achieve of philosophy of the higher education and the country and the level of the researcher adaptation achieved in the ground by meeting need of the states of society and providing with a various scientific and humanitarian specialization. In the extent by the celebrating with this conference success veteran, which is work in the conference communities as well as against time to make the banner of the conference, research the presence of the scale in the scientific knowledge. As called Almighty to keep Iraq there and the secure, rotating and the guarding giving to the civilization of humanity and are on and last prior to the price of the God, Lord of the world, first and last, and may prior to the peace of this messenger and good favor to the over and all the scientific research give the presence of the scientific and the structural and specialization in the higher education, especially in the higher education 10 of the Arab Society of higher Arab Society for stem cell and molecular biology. بسم الله الرحمن الرحيم. يطيب لي أن أرحب بكم في هذا الصرح العلمي البهي ذات الريادة المعرفية المؤتمر العلمي الدولي العاشر للجمعية العربية للخلايا الجذعية والبيولوجي الجزيئي للفترة من 2 إلى 3 نوفمبر 2021 وانطلاقا من دستورنا الإلهي الذي تطرزه آيات الكتاب الحكيم. بسم الله الرحمن الرحيم. يرفع الله الذين آمنوا منكم والذين أوتوا العلم درجات والله بما تعملون خبير. ونحن نباشر مهامنا ومسؤوليتنا في رعاية هذا المؤتمر المعطاء. نضع نصب أعيننا بالمقام الأول تركيزنا على جودة البحث العلمي وريادة الأبحاث وتهيئة الباحثين المشاركين من خلال تنمية مهاراتهم وقدراتهم على التعلم وتسليحهم بالمعرفة ليصبحوا قادة الوطن في المستقبل. يطمح مؤتمرنا أيضا إلى نشر المعرفة وتعزيزها وتوسيع قاعدة الدراسات العلمية، كذلك لمواكبة التطور في العلوم المتقدمة في جميع المجالات والعمل على المساهمة في الاكتشافات والاقتراحات وإحياء حضارة العراق الإنسانية والتاريخ. يحتاج بلدنا اليوم أكثر من أي وقت مضى إلى النهوض بالمسيرة العلمية والتعليمية والبحثية تنفيذا لسياسة وزارة التعليم العالي والبحث العلمي ضمن البرنامج الحكومي الذي أعلنه معالي الوزير في بداية تسنمه الوزارة، حيث أن الدور الملقى على الجامعات وقياداتها لا يقتصر على تزويد المجتمع بالخريجين، بل يتعداه إلى اعتماد المعرفة كمحور أساسي في عملية التعليم والتعلم وإدارة الجودة وضمانها، وتبني المفاهيم الأساسية في جودة التعليم مثل اكتساب المعرفة وإنتاجها ونقلها، فضلا عن تعزيز ثقافة الابتكار والإبداع، وهي تلك الركائز الأساسية التي سنعتمدها في هذا المؤتمر بالتعاون مع الجميع، ومن أجل تكوين جيل جديد قادر على العمل في بناء القاعدة الأساسية والمعرفية التي هدفها الابتكار والتحديث الذي يفضي إلى نهضة العراق. وبناء على ما تقدم فإن المؤتمر العلمي الدولي العاشر الجمعية العربية للخلايا الجذعية والبيولوجيا الجزيئية يهتم بشكل كبير في توفير التربة الخصبة للابتكار والإبداع، وذلك من خلال التركيز على نظم البحث العلمي النوعي والعمل على بناء القدرة الإبداعية والابتكارية للباحثين، وتبني مجموعة متكاملة من السياسات التعليمية التي من شأنها تعزيز التحفيز على الابتكار عند الباحثين والتدريسين في الجامعات العراقية وبقية الجامعات الأخرى من المحيط الإقليمي والعالمي وبالتنسيق بين المؤسسات. وبرغم التحديات التي واجهها العراق بسبب الظروف الصعبة التي مر بها، وما زال يمثل التحدي الأكبر لأقصى الظروف، حيث وقفت الجامعات العراقية شامخة متحدية كل الصعاب برغم قسوة الظروف التي عاشها البلد، إذ خرجت عشرات الآلاف من الطلبة المتميزين في مجال تخصصهم، ولم تتوقف عند هذا بل تسير على وفق اعتماد الجودة للدليل في تقدم المجتمع، كما أنها تسعى الآن إلى تقديم المزيد من خبراتها التراكمية والمعرفية لمواكبة مجتمعنا العراقي ورفده بكافة التطورات العالمية، والإسهام في بناء العراق وبناء الإنسان المبدع والخلاق ومواكبة أجل التطورات الأكاديمية العالمية والتواصل مع حافات العلوم والتكنولوجيا المتقدمة عالميا كي تبقى درسا ونبراسا متوقدا يشع العلم والفضيلة ويرفد الحياة والمجتمع بالإنسان القادر على تطوير وخدمة وطنه وتحقيق طموحات شعبه. في خطوات متسارعة نحو تحقيق الرسالة العلمية، اعتمدت الجامعات العراقية في مشاريع التطوير العلمي اعتمادا لمبدأ المنهاج في التلاقح الفكري والعلمي وتواصلا مع مستحدثات العالم والتكنولوجيا بالتوازي مع الجامعات الأخرى والمؤسسات العلمية الرصينة، وهذا ما اعتمده منهاج المؤتمر الدولي العاشر للجمعية العربية والخلايا الجذعية والبيولوجيا الجزيئي عن طريق اعتماد اللجان المتخصصة في إقامة مؤتمرات وورشات عمل وبحث ومؤلفات من ضمن معاملات التأثير العلمية الرصينة عبر وجود باحثيها ومشاركيهم بما فيه من كل صوب وحدب. وأينما وجد قبس للعلم في هذا الهدف فهو أساسي وكعامل مساعد ونمو كبير لنشر البحوث العلمية في المجلات العلمية الدولية المحكمة واستخراج عدد من براءات الاختراع وتسابقها في علوم الدخول في التواصل بين تصنيفات الهيئات الدولية الجامعية ضمن مبدأ تفاعل الجامعة في المجتمع في سعيها لدعم مؤسسات الدولة ودعم سوق العمل، وهذا ما انتهجه أيضا سياسة ورعاية مؤتمرنا إلى البحث العلمي في هذا المؤتمر البهي. ونبني وإضافة إلى ما تقدم ونختم كلامنا أننا حملنا مسؤولية كبيرة جميعا في الحفاظ على إنجازات البحث العلمي والارتقاء بها بجميع الجهود مشتركة من تدريسين وموظفين وباحثين وطلبة للدراسات العليا لتبقى رافدا تميزا في إعداد الأجيال التي تنهض في سبيل التكنولوجيا والعلوم الحديثة التي توفر الجامعات العراقية بكوادرها التدريسية ذات المراتب العليا إلى الطلبة خلال دراستهم عبر استخدام وسائل تعليمية مختلفة ذات صلة بالتقدم التكنولوجي التي منحت الفرصة للطلبة في تهيئة السبل الأمثل للجوانب النظرية والعملية والتطبيقية بالاعتماد على توجيهات وزارة التعليم والعلم البحث العلمي نحو تبني معايير الجودة والرصانة، وإتاحة الفرصة لإرساء أسس المشاركة في بناء صروح علمية وتربوية تليق بأصالة العراق الحبيب، وهذا ما جعل سعينا نحو التطوير المؤسسي والتنسيق العالي مع جمهورية مصر العربية في إقامة هكذا مؤتمر دولي، والذي لا يمكن أن نحققه إلا بتكاتف أيادي القائمين عليه والمساهمة الفعالة للكادر المتواجد الذي يسعى إلى تحقيق فلسفة العلم على مستوى البلد ليكون ضمن الطموح المتحقق على أرض الواقع، وذلك من خلال تلبية حاجة الدولة والمجتمع ورفدها بالتخصصات العلمية والمختلفة والإنسانية. أوجه تحياتي الخالصة إلى شركاء هذا النجاح العاملين في المؤتمر العلمي الدولي العاشر للخلايا الجذعية والبيولوجيا الجزيئي كافة وفي لجان المؤتمر كافة، وهم يتسابقون الزمن لجعل راية مؤتمرهم هذا تطال الرايات في سماء العلم والمعرفة. أسأل الله تعالى أن يحفظ العراق عزيزا آمنا مشعا بالخير والعطاء والحضارة والإنسانية جمعاء، وآخر دعوانا أن الحمد لله رب العالمين أولا وآخرا، والصلاة والسلام على رسوله دائما وأبدا. وشكرا جزيلا لكم لحسن الاستماع، وأسأل الله أن يوفق الجميع والقائمين على هذا الصرح العلمي في المؤتمر العلمي الدولي العاشر الذي تقومه الجمعية العربية للخلايا الجذعية والبيولوجيا الجزيئي للفترة من 2 إلى 3 نوفمبر 2022. شكرا لكم جميعا.
Thank you, Dr. Basim Khamis Kuti for the support and the cooperations and the good word for the presented. Thank you, Doctor.
Thank you, Doctor. Thanks for all the presence of the committee of the conference.
Welcome. Now, Professor Dr. Abdel Parapans, candidate representative of the Arab Society of Forest Health and Molecular Biology and the head of the Department of Biochemistry at Cairo University. So please, Professor Dr. Abdel Bari.
Assalamu alaikum wa rahmatullahi wa barakatuh. Hello, everyone. On behalf of the Arab Society of Stem Cells and the Molecular Biology and the Organizing and Advisory Committee, we take pleasure in welcoming academic scientists, researchers, students, scholars, and the experts of application fields for the 10th edition of the Era of Biotechnology and the Personalized Medicine Conference to foster the progress in the field by contributing with your expertise to what promises to be a very comprehensive and exciting meeting. International Conference on Biotechnology and Personalized Medicine, which is held during this November 2 to 3, 2022, is interdisciplinary platform for the presentation of new advances and research results in the field of biotechnology and medicine. The event will have a plenary speakers, established keynote speakers, active invited speakers, and the fresh contributed speakers. In addition, variety of presentation along with workshop and the special session will be interested in audience. The aim of the 10th edition 2022 is to promote quality research and real-world impact in an atmosphere of international cooperation between scientists by bringing together again the world-class researchers, international communities, and industrial heads to discuss the latest developments and innovation in the field of biotechnology and medicine. We sincerely hope that the 10th edition 2022 serves as an international platform for meeting researchers from around the universities, widen professional contact, and create new opportunities, including establishment, new collaborations. We are looking forward to seeing you again and again. And the young researchers' opportunity, we make sure to support and motivate young researchers by establishing their academic and professional relationships, improving their morale and the confidence of presenting research in an international platform, interacting with expertise in their respective departments, clearing your inhibition of adjusting to foreign environment, providing holistic experience of an academic tourism. Finally, I would like to express my gratitude to the Iraqi partnership. This is the second time that we have a partnership at the Arab Society and the STEMS and the Molecular Biology. And I would like to express my special thanks to Dr. Haider Al-Saidi for organizing and preparing this event. Thank you very much.
Thank you, Dr. Abdel Bari Prince, for the good words and cooperations of the conference. Now, my colleague in the preparatory committee is Dr. Ali from the medical college from the West University. Please, kindly, Dr. Ali.
Assalamu alaikum and good morning, everyone. Thank you, Dr. Haller, for this word. Now I would like to invite the keynote speakers. We will start with the international keynote speakers, Associate Professor Dr. Chwali Swan from University Technology of Malaysia, Metabolites Profiling Laboratory, Institute of Bioproduct Development, University Technology Malaysia, and Here topic concentrating rosmarinic acid rich extract from orthosphone erystatus from the for anti-hyperglycemia. Dr. Chua.
Okay, thank you, Dr. Ali. I will share my slide first. Can you see my slide?
Yeah, participants, the slides clear?
I try to make presentation mode.
Yeah.
Okay, can you see?
Yes, already presented, yes.
Okay, thank you. Okay, good day and good morning, everyone. First of all, I would like to thank the organizer to invite me as the keynote speaker for this conference. And special thanks also to Dr. Ali, who is my ex-student in Malaysia. He has put a lot of effort in coordinating the conference and make the conference as a success. So thank you very much. The topic of my today's presentation will be focused on concentrating rosmarinic acid rich extract from orthosphone erystatus for anti-hyperglycemia. So, orthosphone erystatus from the family of Lamiaceae is a traditional herbs in Malaysia. This herbs is popular in Asia region. Many people use the herbs as a herbal drink and as eggnog medicine for many diseases. This is because the herbs rich in polyphenol, the bioactive compound. One of the major bioactive compound in these herbs is rosmarinic acid, which consists of 53 to 299 milligram per gram, according to the literature. So this is the molecular structure or chemical structure of the compound, consists of two major components, the unit of caffeic acid, and the other one is dihydroxyl phenylactic acid component. This compound has been reported to have this pharmacological importance, especially in antioxidant, anti-cancer, anti-diabetic, and many more properties. That is why we can see that many products derived from this rosmarinic acid extract available in the market. As you think, we have health supplement from rosmarinic acid extract. Usually people get this health supplement to extract the bioactive compound from rosemary plant. But in fact, from our research, we found out that our orthosphone erystatus got higher content of rosmarinic acid compared to the rosemary. Therefore, it's more economical value if you use the orthosphone to prepare rosmarinic acid for the health supplement product in the future. So in today's presentation, I'm focusing on the topic to use rosmarinic acid, or I call it RA here as an inhibitor to inhibit two major enzymes contribute to type 2 diabetes mellitus, which is alpha amylase and alpha glucosidase. So in this case, we are investigating the role of rosmarinic acid to prevent the excessive glucose level in the blood. So the objective of the study are to compare the efficiency of column chromatography, the first technique, column chromatography, or we call it CC, and the second technique, liquid-liquid extraction integrated with solid phase extraction technique, or we call it LLE-SPE, in concentrating rosmarinic acid from Orthosiform aristatus. And the second objective would be to determine the kinetic mode of inhibition by rosmarinic acid against alpha amylase and alpha glucosidase. This is the overall methodology flow of our work here. As we can see at the beginning, we use reflux technique to prepare the crude extract from the raw material of the plants. From the crude extract, we further fractionate it in order to harvest or recover rosmarinic acid from the crude extract. Here we use two methods, the method 1 and the method 2. The method 1, we call it CC or column chromatography, and then the method 2, we call it LLE and SPE. In the method 1, we have to choose the best solvent, the most suitable solvent to prepare and elute the compound from the crude extract. Here we use thin layer chromatography, just a simple method for solvent selection. And then we also optimize our pet bed column to harvest rosmarinic acid in high yield. We change the pet bed height from 16 centimeter to 32 centimeter in order to achieve or to get the highest yield of target compound fractions. So in the second method, we use LLE for solvent selection. A few choice of solvent we apply. We're going to investigate which solvent system is the best to get higher yield of rosmarinic acid. Subsequently, we use the sample for SPE process. Fraction it into individual fraction to harvest or to recover rosmarinic acid in high yield. And then from these two processes, we get the sample for quantification and identification using LC-MS-MS. And then at the last part of the study, we use the enzyme we use rosmarinic acid to inhibit the enzyme, the two enzymes, and then we investigate the kinetic profile of the enzyme inhibited by rosmarinic acid. This is the overall methodology flow of my work for today's presentation. As this is the slide for the result and discussion, as you can see that at the upper layer, we have the chromatogram for the crude extract. As we can see that rosmarinic acid has the highest component in the crude extract. So for identification, we confirm that the compound belongs to rosmarinic acid because we can get the molecular ion of the compound, and then we are also matched with the database that this compound has the product ion we require in the fraction. So here we have the result for thin layer chromatography. For thin layer chromatography of solvent selection, here we apply binary solvent system, ethyl acetate and ethanol at different concentrations. And then we found out that with the addition of 0.1% formic acid, we are able to get a nice band for rosmarinic acid with the appropriate retention factor we require in the intermediate. We cannot we cannot get the band at the high part or the lower part of the thin layer chromatogram. We have to choose the solvent system which is able to give the retention factor in the intermediate so that we later on we can apply the solvent system to fractionate the crude extract to harvest rosmarinic acid in high yield. So this is just this chromatogram, thin layer chromatogram is just a comparison to see that without addition of formic acid, we have this stirring effect. We cannot get a very nice separation or purification for the rosmarinic acid. So again, since this is the optimum range for the thin layer chromatography, optimum solvent system will be from 60 to 100% ethyl acetate. So we are going to apply this Sowen system in the following step to pack the column chromatography for fractionation. This is the system. We pack it in the lab. And then, as mentioned just now, we prepare the Sowen system for 60% to up to 99.9% of ethyl acetate because we got 0.1% of formic acid added inside the Sowen system. So from here, we can notice that the Sowen system of 70% ethyl acetate is the best Sowen because we just require minimum volume of Sowen to harvest the rosmarinic acid from the crude extract. And then again, from the fractionation system, we collected a lot of fraction from 1 to 20 fraction. And then we found out that the fraction number 8, for this study, I focused on fraction number 8. The other fraction we use for other purpose because the purity of rosmarinic acid is not so pure. Even though they have high concentration of rosmarinic acid, but not so pure. That's why we use for other purpose. For today's presentation, I'm focusing on fraction number 8 because we're able to achieve 100% pure rosmarinic acid from the fraction, even though the recovery percent is just 15.6% in this case. So based on our optimization for the packed column height, I raised the packed column height from 16 centimeter to 32 centimeter column height, and then the result found out that the purity are not much changed, and as well as the rosmarinic acid recovery also not much change. That's why we maintain the minimum height for this packed column. If you would like to get higher yield, maybe you can increase your size of your column. So now we come to the second method, LLE and SPE, to collecting rosmarinic acid rich fractions. In this case, we use 3 different types of Sowen system for fractionating the crude extract of the herbal plants. So the result found out that the second Sowen system is the best to be used in this case because rosmarinic acid contains the highest amount in the ethyl acetate phase. The use of diethyl ether is with the purpose to remove all the lipid substance as a impurity. Majority of the target compound, bioactive compound, RA or rosmarinic acid are present in the ethyl acetate phase. So subsequently, we use the sample for SPE fractionation. This is the SPE system set up. Saying in this case, we collected 6 fractions, and then we analyzed the sample using HPLC. The chromatograph of the HPLC, we decided to convert fraction number 1 until number 3. So the found out that the purity of rosmarinic acid in this case is just 75%, and the recovery is 76% recovery. So as a conclusion for the method to prepare rosmarinic acid rich fraction or extracts, we prefer column chromatography method, the first method, because from the fraction number 8, we can get high purity, which is 100%, and high yield compared to the other methods. So column chromatography use silica gels as a polar adsorbent for this fractionation work. And then whereas the SPE use the non-polar adsorbent for the fractionation, for the clean up process. So now we come to the second part, second objective of the study. Using the sample we prepare from the previous method 1 and 2 to analyze the activity of rosmarinic inhibit our 2 target enzyme. The first one is alpha amylase. Alpha amylase, we try to inhibit it to prevent it converting the starch into maltose. From the result, we see that the rosmarinic sample with concentration 62.5 milligram per ml, we can achieve almost 100% inhibition the alpha amylase. And then we also can see from the bar chart here, the crude extract has the lowest performance compared to the sample we prepare from chromatography LLE method. So, this fractionation technique helpful to semi-purify rosmarinic acid in the fraction so that it can higher activity to inhibit the alpha amylase. For better, we express the result in terms of effective concentration of inhibition in IC50 for alpha amylase. As we all know, the lower activity of IC50, better inhibition capacity. So from the bar chart here, as we can see, the sample we prepare from the method 1cc column chromatography have comparable performance with the positive control drug alkabose, and as well as with the standard we purchase from the supplier. And then the crude extract has the lowest, the poorest performance inhibiting the alpha amylase. So fractionating the rosmarinic acid from the crude extract actually is helpful to increase the inhibition capacity of the sample. So this is the kinetic profile for rosmarinic acid as an inhibitor to inhibit alpha amylase. In this case, from the plot, the Michaelis-Menten plot, we notice that the rosmarinic acid acting as a competitive inhibitor. It competes with the substrate to fit into the active site of the enzyme for the process. So by fitting into the enzyme active site, the enzyme will be deactivated, and then the enzyme cannot convert the starch into maltose anymore. This is the way how rosmarinic acid performs. So from the curve, this kind of competitive inhibition mode, rosmarinic acid did not change the maximum rate of reaction. The maximum rate of reaction maintained almost the same. They only shifted the Michaelis-Menten constant from 3.3 millimolar to 4.1 millimolar. This is the constant to express the affinity of rosmarinic acid against the alpha amylase. For the second enzyme, alpha glucosidase inhibition activity, we also noticed that only 10 milligram per ml of the sample are sufficient to inhibit alpha glucosidase about 100% of inhibition. So again, from here, the bar chart, we can see that the sample with the red color we prepare from the method 1 column chromatography have better result compared to the acobase, which is the positive control drug, better than the drug performance in inhibiting the glucosidase activity. So the rosmarinic acid is a good inhibitor to inhibit glucosidase enzyme. So for effective comparison, we also plot the result in terms of IC50, and then again we can see that our sample from the first method of fractionation comparable with the standard we purchased and better than the positive control drug acabose, and then better than the crude extract as well as sample prepared from the second method. So, but in this case, the compound, the rosmarinic acid compound, behave in non-competitive inhibition mode. The profile will be slightly different from the alpha amylase. We can see from here that with the inhibitor, rosmarinic acid inhibitor, the reaction rate will be lower, whereas the Michaelis-Menten constant remain the same. This is how rosmarinic acid react differently with different enzymes. here with the non-competitive inhibition mode, the rosmarinic acid did not compete with the substrate to the enzyme active site. Instead, it bind to the other side and then make the inhibited for the subsequent process to convert maltose into glucose. So here we come to the conclusion part. And then as a conclusions, we choose column chromatography method in fractionating rosmarinic acid from the crude extract of the herbs Orthosiphon aristatus, which is a common herbs used in Malaysia because of its beneficial and many remarkable biological activity. So the best solvent system in our study is 70% ethyl acetate, 29.9% ethanol with 0.1 formic acid. And then it could increase the concentration of rosmarinic acid from 3.6% in crude extract to 100% in the fraction number 8. The yield is also increased to 4.5%. So for the enzyme kinetic part, we conclude that both enzyme inhibition activity better than crude extract. We prepare the rosmarinic acid fraction-rich extract, which can show better activity than the crude extract to inhibit these two enzymes. And then last, what is the interesting part is that rosmarinic acid actually is a very good inhibitor compared to the acarbose, which can inhibit the enzyme in lower concentrations compared to the positive control drugs just now. And then out from our work, from this work, we managed to publish 2 articles, international paper. One is in current enzyme inhibition activity, and the other one is about the technique to prepare the rosmarinic acid-rich extract from Orthosiphon in Journal of Liquid Chromatography and Related Technique. That's it for my presentation. Thank you very much.
Thank you so much, dear editor, for this precious topic. Now we have 5 minutes for discussion, so if there are any questions, please raise the hand. Any question, please? Okay, thank you so much, Dr. Now we will start with the second keynote speakers, Associate Professor Dr. Ilyas Said from Department of Microbiology and Immunology, College of Medicine and Health Science, Sultan Qaboos University. His title, The Cytokines Midkine Links Immunity and Angiogenesis.
Thank you, Dr. Ali, for introducing me. I'll start by sharing my screen. Is the voice clear to you? Hello?
Okay, Dr. Clear, Dr.
Yeah.
Clear.
Oh,
okay,
yeah,
go ahead, please.
Sure. Okay. On my screen, I have it in full screen, but let me just share the whole screen. Just 1 second, please. Sorry for this. Okay, so thank you, Dr. Ali, and thank you for all the organizers for this invitation. I'm going to talk to you today about the cytokine and growth factor midkine and how it links the immunity, and precisely the innate immunity, with angiogenesis. So to start, I would like to introduce what midkine is. Midkine is a heparin-binding growth factor, and it is considered as a cytokine. It wasn't considered as a cytokine in the beginning, but now it's been almost 20 years that it has been considered as a cytokine. It is a 13 kilodalton protein that has 143 amino acids, and it's non-glycosylated. It has cysteine and basic residues, and it has 5D sulfide bonds. It consists for its activity of mainly an N-terminal domain and a C-terminal domain, and it belongs to the midkine pleiotrophine family of cytokines. So this family of cytokines is formed of two cytokines, which are midkine, MK, and pleiotrophine, PTN. There were many receptors that were identified for midkine, such as anaplastic lymphoma kinase, cell surface nucleolin, LRP, RPTP, neuroglycans, neuroglycan C, Notch2, and integrin. And actually, we have previously identified nucleolin, the cell surface nucleolin, as the low affinity receptor for midkine. So midkine has clearly two binding curves, one of a high affinity and the other one with a low affinity. And this low affinity receptor was identified by us to be the cell surface nucleolin. Why we are interested in midkine? It's because midkine has many involvements in different physiological processes, such as tissue repair, proliferation of stem cells, inflammation, innate immunity, angiogenesis, development, reproduction, control of blood pressure, embryogenesis, and neurogenesis. It is able to regulate some cells of the immune system, such as macrophages, to activate CD4 T lymphocytes and suppress regulatory T cells. For its effect on CD8 T cells, then we have contradictory information between the different literature. Some report a stimulating effect, and others report an inhibitory effect. And it has also a survival, a pro-survival effect on B cells. Midkine is also implicated in diseases such as autoimmune diseases, including rheumatoid arthritis, systemic lupus erythematosus, SLE, and multiple sclerosis. Now, there is a huge evidence that midkine is produced by multiple types of cancer cells, and it promotes the tumor cell proliferation, and it promotes the angiogenesis also that is needed for these tumor cells to grow. And it also promotes, for instance, hepatocellular carcinoma cells proliferation. We have shown that midkine can inhibit HIV binding to the cells. And actually, midkine inhibits the binding of HIV isolates to T cells. And this inhibition is mediated by the interaction of midkine with the cell surface nucleolin, as we've shown that this happens at the same concentrations at which midkine binds to nucleolin. We also showed that not only midkine can inhibit HIV binding to the cells, but also PTN pleiotrophine, which is the brother of midkine that forms together with him the same family of cytokines, also can inhibit HIV binding by binding to the cell surface nucleolin. So midkine has an important role in angiogenesis, and this role is very important because angiogenesis plays a vital role in cancer. And we also know that the proliferation of endothelial cells is very important for angiogenesis. And actually, it was shown that breast cancer cells, so breast carcinoma cells that are constitutively expressing midkine, will give a growth advantage will have a growth advantage, and this is probably because of the angiogenic activity of midkine. And also, it was known that the addition of midkine to endothelial cells is going to induce their proliferation. Now, for the expression of midkine by innate immune cells, there were some reports about MITKINE expression in monocytes in amphibians and axolotl, and in macrophages in rabbits and mice. In humans, there were no such reports, but in monocytes, there was an expression of MITKINE in monocytes under hypoxic conditions. And in the cell line THP1, which is macrophage-like cell line, the expression of MITKINE is controversial. So no study showed the expression of MITKINE by human macrophages or dendritic cells that are primary cells in any species for dendritic cells and in primary macrophages in a human. So where our questions were, is MITKINE produced by human innate antigen-presenting cells, monocytes, dendritic cells, and macrophages? And if yes, how does that influence endothelial cells? So we first stimulated monocytes that we isolated from the blood with lipopolysaccharides, LPS, and we observed that MITKINE is produced at the messenger RNA level, but also at the protein level. We differentiated those monocytes in vitro to macrophages and monocyte-derived dendritic cells and stimulated them again with LPS, and we saw that MITKINE is produced at the mRNA level and also at the protein level, both in by macrophages and monocyte-derived dendritic cells. Now, we also isolated circulating dendritic cells from the blood. So we isolated plasmacytoid dendritic cells and myeloid dendritic cells from the blood, and we stimulated them again. Now, this time for PDCs, we stimulated them with resiquimod, not LPS, because they do not have TLR4, which is the receptor of LPS, but they have the TLR7, which is a receptor for resiquimod, which mimics the single-stranded RNA activity. And we found that plasmacytoid dendritic cells did express MITKINE at the mRNA level and at the protein level when stimulated via their TLR7. However, this was not observed in myeloid dendritic cells. So under no conditions, myeloid dendritic cells did produce MITKINE when they were stimulated, although the stimulation was effective because these cells increased the levels of the co-stimulatory molecules CD80 and CD86 on the surface when they were stimulated with lipopolysaccharide. We then looked at the expression of MITKINE in vivo. Is it expressed really in macrophages and dendritic cells in vivo? So we had stained biopsies of a human tonsils that were showing follicular hyperplasia. So the brown color is the MITKINE antibody. And you can see here by morphology that we have very nice shapes of dendritic cells and here macrophages that are producing, that are positive for MITKINE, so they are producing MITKINE. So we wanted to make sure that these cells are really macrophages and dendritic cells, and for that we used double immunohistochemistry staining. So our first antibody was directed to MITKINE, specific of MITKINE, and the other one was directed towards one of the markers of either MDDCs, PDCs, monocytes, and macrophages. So for MDDCs, we could use either CD1C or CD11C, but CD11C is a better marker in vivo than CD1C. So we chose CD11C. However, it stains also some subtypes of dendritic cells and macrophages and monocytes. For plasmacytoid dendritic cells, the choice was CLEC4C, which is also called CD303, because it is a specific marker of these plasmacytoid dendritic cells. For monocytes and macrophages, we have a choice between CD68, CD14, and CD163, and also CD16. They are also expressed on some disease subsets, but the issue with CD14, CD163, and CD16 is that their level can be modulated by activation. So you might miss it or amplify what you are seeing depending on how activated your cells are. So we used CD68 in this situation to stain these cells. So our markers were anti-CD11C, anti-CLEC4C, and anti-CD68. As I said, this was together with midkine. So in these images, you can see that midkine staining is shown in is actually brown. The color was brown, and for the subset marker, the color was red. So again, those are biopsies of human tonsils demonstrating follicular hyperplasia. And you can see clearly that CD11C positive cells here in red, they do also stain for brown, which is midkine. So they are expressing midkine in vivo. This also is applicable to the CLEC4C positive cells. So they are plasmacytoid dendritic cells. You can see the red staining on these cells and the brown staining in the same cells. We also detected CD68 positive cells that are also positive for midkine here, as you can see in brown. So we concluded that in vivo CD11C positive cells, CLEC4C positive cells, and CD68 positive cells do produce midkine. Now, the percentage of these cells that are producing midkine, so CD11C, CLEC4C, and CD68 positive cells, was very similar. So if you take the percentage of cells that are producing midkine among these cells, it's very similar. However, when you take the cell count in the biopsy, the CD11C cell count in total, regardless of their expression of midkine, was the highest, followed by the CLEC4C and then the CD68 positive cells. So actually, if you take a number, the positive cells that are really producing midkine, then you will have still the CD11C higher and CLEC4C higher, but the lowest will be the CD68 positive cells. Now we show that in vitro and in vivo and ex vivo, the innate immune APCs can be producing midkine when they are stimulated. And we wanted to know how this production is occurring and what is the link between the toll-like receptor stimulation and midkine production in these cells. Actually, it is known that the midkine gene has an NF-kappa B binding site in its 5' non-coding region. So we use an inhibitor of NF-kappa B P65 translocation, which is GSH23, to see if we will be able to block the production of midkine by these cells. We stimulated total PBMCs, peripheral blood mononuclear cells, or macrophages or dendritic cells, monocyte-derived dendritic cells, with 20 and 40 nanograms of LPS in the presence of GSH or the absence. And we saw that the presence of the inhibitor of NF-kappa B translocation, or the P65 subunit translocation, did inhibit the production of midkine by these cells. And as a control, we observed also that TNF-alpha, which is known to be under the control of NF-kappa B and produced because of the triggering of toll-like receptors, was also inhibited in these cells by GSH23. So GSH23 inhibited the production of midkine, and it is known to be an inhibitor of NF-kappa B. So now we know that NF-kappa B plays an important role in the production of midkine that is stimulated by the triggering through toll-like receptors in the innate antigen-presenting cells. Another enzyme that could potentially be implicated in the production of midkine in these cells is PDE4, and we stimulated the cell and put in the presence of roliprime, which is an inhibitor of PDE4, and found that PDE4 can also inhibitor can also inhibit the production of midkine by these same cells. So this suggests also a role for PDE4 in the production of midkine upon TLR stimulation. Now, we know that from the literature that soluble midkine is able to stimulate the proliferation of vascular endothelial cells. And we know now that midkine is produced by macrophages and monocyte-derived dendritic cells. And also the literature tells us that these cells can modulate the proliferation of endothelial cells. So we wanted to see if midkine produced by macrophages and monocyte-derived dendritic cells is mediating this ability of these innate immune cells to modulate the proliferation of endothelial cells. We use siRNA directed toward midkine, and we show here that this siRNA abrogated completely the production of midkine by macrophages and monocyte-derived dendritic cells when cells were electroporated in the presence of this midkine-specific siRNA. We use the supernatant to assess the proliferation of the endothelial cells. I want just to introduce the proliferation test that we use, just in case you're not familiar with. So we use a staining with CFSE. The CFSE is a dye that will bind in a non-specific way to proteins in the cytoplasm of the cells, and it will it's a green fluorescent stain that is observable by flow cytometry. Now, as the cells divide, this CFSE is going to be diluted with each division. So with each division, we are going to lose in intensity of the CFSE. So the less intensity you have, the more that your cells have proliferated, as you can see here as an example. So here are non-proliferating cells. As the cells are proliferating, they are inheriting less amount of CFSE, and you can observe the proliferation. So we stained these, endothelial cells with CFSE and incubated them in the presence of supernatant of non-stimulated macrophages or monocyte dendritic cells, or stimulated monocyte and dendritic cells in the presence or absence of the midkine-specific siRNA. We observed that, as you can see here, that the inhibition of midkine production by monocyte-derived dendritic cells and macrophages did affect the proliferation of the endothelial cells and actually inhibited the enhancing effect of the supernatant of macrophages and monocyte-derived dendritic cells on the endothelial cells. You can see here with the CFSE that when you have the endothelial cells alone, they do proliferate, but you still have cells that are not proliferating much. When you put the supernatant of the macrophages or dendritic cells, you lose the population that did not proliferate much, because all the cells proliferated at stronger level. But when you have and when you inhibit midkine production, you gain these cells again because they do not proliferate as much as with the total supernatant of macrophages and dendritic cells. This was rescued by the addition of midkine to show that it's really specific to midkine. We did not observe effects of this MITKINE inhibition on the viability of the endothelial cells as observed by annexin 5 and 780 staining. So in conclusion, we show that MITKINE is produced by human plasmacytoid dendritic cells, monocytes, and their derived macrophages and dendritic cells. However, myeloid dendritic cells do not produce MITKINE, and that the TLR-induced MITKINE production is mediated by NF-kappa B, and that there is a potential role for the PDE4 molecule in this MITKINE production. And finally, that the MITKINE produced by macrophages and monocyte-derived dendritic cells, because of the triggering of toll-like receptors, can control and enhance the proliferation of endothelial cells that are very important in angiogenesis. So the results of this presentation were published in this article, so you can use it as a reference for the information that I've been telling you and also the background information that I gave you. And finally, I would like to thank all the audience and also all our collaborators in the different departments and Sultan Qaboos University. So our department, the Microbiology and Immunology Department, the Department of Pathology, the Hematology, and also Biochemistry and Biology in Sultan Qaboos University, and the Department of Medicine and the AFH, and also the Department of Medicine and Lausanne University. Thank you very much for your attention.
Thank you so much, Dr. Elias, for this important lecture. And now we will start we will open the time for questions. So please, if there are any questions, raise your hand. Any question, please? So now we will start with the third keynote speakers, Professor Dr. Tahir Harami, Program Director of General Surgery in Kurdistan Board for Higher Medical Specializations. This topic title, Pre-Circle Dermolysis, a Case Report. Prof. Dr. Tahir.
Good morning. Thanks a lot, Dr. Ali, and thanks for all. I was wishing to meet you in Baghdad, inshallah, for the next time. My case is a case report just talking about pre-sacral dermolysis, which had been presented to me. This is a very rare condition. It will be found in 1 in 40,000. In the sacropoccygeal area lies between the upper two-thirds of the rectum and the sacrum above the rectosacral fascia. It is bounded by the rectum anteriorly, presacral fascia posteriorly, and endopelvic fascia laterally. Contains multiple embryonic remnants derived from a variety of tissue like neuroectoderm, notochord, and hindgut. Either they will be solid or cystic, but cystic are more common. Mostly they will affect and present in the middle-aged woman. Female to male ratio is 10:1. Often asymptomatic, symptoms resulting from local mass effect, for example, constipation, fullness, lower abdominal pain, dysuria. They will be palpable retrorectal mass at digital rectal examination. Diagnosis is challenging because of unusual presentation and high level of suspicion required for their diagnosis. Digital rectal examination is a cornerstone of diagnosis and has about 97% sensitivity. Topographic assessment and anatomic relation to other structures will be evaluated by transrectal ultrasound, tomography, and MRI. Pelvic MRI is the most sensitive and specific. The treatment is almost always surgical, either sacrococcygeal or transabdominal, or sometime and very rarely combined abdominal sacral approach by the needed. But the sacrococcygeal approach for complete excision, minimum mortality. Our patient, we did for him, all the patients really, complete resection, smooth postoperative period, and no recurrence recorded in the follow-up period. Mechanical and chemical bowel preparation needed for all the cases of the surgery. They may be resected either under general anesthesia or spinal. The area of proposed incision should be marked in the prone position through elliptical incision with the excision of the coccyx and exploration of the presacral region. Complete excision of the tumor is necessary in order to prevent recurrence. Postoperatively, after excision of the tumor, the wound left open for the healing by secondary intention. Initially, the wound should be packed with iodine-soaked packs for 2 days. After 2 days, the pack should be removed and daily dressing with normal saline is applied. This is really the paper of the 9 cases we did sacrococcygeal approach for them and will be published. The dermoisis of our case, this was an amazing case, a 48-year-old housewife female, gravida 3, para 2, abortion 1, both cesarean section, and past medical history. She's a known case of diabetes. Past surgical history, she had a history of 2 cesarean section, once midline laparotomy for the same condition, 1 DNC suspecting that there is something abnormal in the uterus, and 3 laparoscopic surgeries for drainage of the same pathology. 17 times her pathology had been drained as an abscess per vaginal. 10 times the same abscess had been drained per rectal, and she had underwent hysterectomy in 2020, thinking that the origin of the abscess is the uterus. In 1996, she had an emergency C-section for her first child, followed by wound infection. 6 months later, severe pelvic pain, abdominal distension, constipation, dyspareunia, and diagnosed as pelvic abscess and vaginal discharge. She didn't cure. Symptoms continued and interfered with her daily activity, aggravated by walking and standing, relieved by lying down and hot pads sometimes. Few months later, she became again pregnant, followed by abortion. During dilatation and curettage, they found a mass. They did for her laparotomy and resection of some tissue. 2 months later, the symptoms recur every 3 to 6 months, followed by drainage. Again, longest time 1 year between each 2 operations. Used to have multiple recurrences and drainage per vaginal, per rectal, phalangeal incision, midline, laparotomy, laparoscopically. Twice spontaneous rupture of the cyst had been occurred per vagina, and she was suffering from defecation. Sometimes per rectal drainage also was occurring. In 2020, she underwent subtotal hysterectomy and resection of relation of the cyst to uterus, but symptoms still were there. One of the surgeons in one of the governorate of Iraq, because this case, she was really from Mosul. He gave her this what's called a map that the abscess is between vagina and the uterus, and when she is going to any surgeon, she should give it to him in order to be able for the drainage. Clinical examination, digital rectal examination, there was a cystic mass protruding from the left side of the pelvic, pressing on the rectum. And I did for her pelvic MRI. There was a 70-60 by 65 by 60 millimeter size multiloculated cyst centered within the posterior pelvic compartment of the left side of the midline, behind the lower rectum and anal canal, extending to the anococcygeal raphe, appears to be attached to fibers of the left levator ani. It has thin, enhanced wall and 30 short peripheral restricted diffusion wall and foci enhancing component. Overall picture in favor of tailgate duplication cyst compresses and displaces the anal canal and lower rectum to the right side. This was the MRI of the patient, and this really, if you look at this anterior abdominal wall, and this is a cyst pressing on the rectum and vagina. Operative notes, we gave her spinal anesthesia in a prone position. We did sacrococcygeal approach, a cyst found attached to the coccyx directed toward the vagina and the rectum from the left side, not communicating with it. A lot of granulation tissue found due to multiple drainage, coccyectomy with separation of the coccyx from the surrounding structure, cyst removed with repair of the posterior vaginal wall because so many times it had been raped and the cyst was communicating with the posterior vaginal wall. The wound left open for healing by secondary intention and packed with iodine-soaked packs. Packs removed after 2 days later, daily dressing done with normal saline and discharged with no complication a few days later. Histopathological examination comes back as a dermoid cyst. Really, regarding the discussion of the case, it's a rare case, many asymptomatic, often found on physical examination or imaging. Our case misdiagnosed from the beginning, presented with vague pelvic pain, abdominal distension, constipation, diagnosed as pelvic abscess, underwent many operations for drainage and difficult approach. Congenital tumors are most common in this area. Most of them arise from the 3 germinal layer, more common in females, may arise from ectoderm, dermoid, and epidermoid, sequestration of the hind gut remnants, tail gut and rectal cysts, defects in the neural tube, anterior and sacral meningocele, tumor-like chordomas may affect this area. Classified into 3 groups, most of them are benign, containing well-differentiated tissue, immature without malignant feature. Some of them may be malignant. A careful digital rectal examination will diagnose the case in 90%. Appropriate diagnosis needs for these cases, primary and only satisfactory treatment will be complete surgical resection. 3 approaches commonly: abdominal, transsacral, sacrococcygeal, and combined abdominal sacral approach. Sacrococcygeal associated with lower blood supply. In these cases, really detailed history and complete physical examination is mandatory, and it will help to reach a diagnosis. Comprehensive clinical assessment, including rectal examination, is essential to establish diagnosis. Contrast enhanced MRI of the pelvis is the gold standard investigation and the most sensitive and specific investigation also. But needle biopsy is not good. Biopsy for adjuvant or palliative therapy in case of malignant cases for unresectable lesion may be needed. For example, in case of chordomas, significant comorbidities that preclude pelvic surgery associated with these cases. A conclusion, the diagnosis is uncommon. Diagnosis require high level of suspicion along with digital rectal examination. Sacrococcygeal approach as really will help for complete resection and associated with minimal morbidity and mortality, less blood loss, less rectal and adjacent organ injury, and the incision is very good regarding the cosmetic result of this case. Digital rectal examination is needed, and the aphorism of that, if you are not putting to your finger, you should put your foot, really applied in these cases. And all cases of pilonidal sinus or abscesses in the anal cleft should undergo digital rectal examination to exclude presacral lesion. Really, I had operated on a case 11 times. She had underwent operation for pilonidal sinus, and she was a case of presacral dermoid. And this is really the video of our last case. If you had this is perioperative, this is the incision presacral, and this is the fluid of the sac after trying to extract it. After excision of the coccyx, all this fluid was coming out from the presacral sac, which she had underwent 38 operation for this condition, and all of the procedure were done either laparoscopically or transabdominally or vaginally or rectally or self-drainage through the rectum and the vagina for the same case. As I mentioned, really, we did the procedure under spinal anesthesia after preparation of the bowel, because there is a chance of injuring of the rectum, and if it's prepared, we could really suture it, and you should take informed consent for the patient regarding the possibility of colostomy, temporary colostomy. Till now, we did about 16 cases of these the problems. Everyone with a diagnosis, and one of them, he was suffering from a presacral hydatid cyst, because hydatid cyst, as mentioned in our books, and we had been taught by our seniors, really may affect any tissue in the body, including presacral. And these are our references. Thanks a lot. Any question or comment?
Thank you so much, dear Doctor. Actually, it's a very interesting topic. So if there are any questions, please raise your hand. Any question, please? Thank you so much, dear Doctor. Now we'll go to take a rest 15 minutes. Assalamu alaikum and good morning, everyone. Now we will start with the keynote speaker, Dr. Hina Al-Mahmoudi from College of Biotechnology, Molecular and Medical Biotechnology in Nahrain University. Her topic is the zebrafish xenograft model for investigating cancer cells migration and metastasis. Please start, Doctor.
Good morning, everyone. Thank you, Dr. Ali, for your introducing myself. My presentation, it will be a talk about zebrafish xenograft model for investigating cancer cells, migration, and metastasis. Firstly, I will apologize about my voice. I had cough, so forgive me about that. The headlines of my talk, it will be introduction, advantages of zebrafish, and also we will mention about zebrafish as a model to study human cancer, and also the methods for studying invasion of cancer cells by using zebrafish as a model. And also the technique will use a microinjection technique for this experiment. Firstly, zebrafish dinorrhea is a common and useful scientific model organism for studies of vertebrate development and gene function. It is used as a laboratory animal model. By the way, this model, it is new. It was start using this model for cancer research at 2007, and nowadays it will be used for a wide experiment. The geographic range of this model separate in the east of Asia, as we see here with red dot, and the size of adult of zebrafish is around 2 to 5 centimeters. But actually, we use embryo for our experiment. There are many advantages of using zebrafish as a model for cancer studies. Firstly, rapid development of this type of animal, where fish can produce hundreds of eggs a day. Also, the most important things in this type of model, visual transparency. Zebrafish can be exploited further by use of fluorescent dyes to label cells and visualize invasion and metastasis. Easy of keeping zebrafish due to low cost and small size of zebrafish compared to other animals models like mice and rats. The circularity system of zebrafish like that of mammals, so that very important because we need to use a model looks like mammals. Also, there is a 70% similarity in genetic homology in zebrafish and human. Nowadays, zebrafish dinorrhea models become a popular model to understand many things in cancer studies. Tumor proliferation, ingenogenesis, invasion and metastasis, and this what I will focus on it in my talk. Also, crosstalk between cancer and host cells. As we know, cell invasion is related to cell migration and defines the ability of cells to become motile and to navigate through the extracellular matrix within the tissue or to infiltrate neighboring tissues. And also cancer cells that become invasive may disseminate to secondary sites and form metastasis. Actually, this is the main problem for progression of cancer and become aggressive. So we need more knowledge about how cells can be ability to disseminate from site to another site. And we need a new technique and a new model to understand this process. As we know, metastasis can be defined as a dissemination of cancer cells from the primary tumor to a distant site. It is multi-step and inefficient process, but is the main cause of death in cancer patients. Okay, in this case, how can study the migration of cancer cells? Cancer cells can be moved or disseminate from a primary site to another site. To investigate this ability, okay, to investigate this ability for movement, we need a new model. And to follow these cancer cells when it become disseminate to site from site to another site. Previously, they used Boyden chamber, transwell chamber for in vitro, but actually we need some new things for investigate that in vivo. In this model, in this type of experiment done inside the lab, these Boyden chamber include two chambers. The first one injects cells in this layer. If cells have ability to move, it will go through these pores to another chamber. So these cells can be moved from side to another side. This cells has ability for movement. As we mentioned, this type of experiment doing in vitro. Okay, what about in vivo methods for studying cancer cell migration? We need technique and models. In the past, they use injection, normal injection for different types of models. But in our experiment, we did macro injection, this type of injection done under microscope. And for injection, they use mice as a model, also rat as a model for injection. But for microinjection, we use new model, which is zebra fish. As we mentioned in this presentation previously about the advantages to use this zebra fish compared to another type of models like mice and rats. So Firstly, we start labeling cells for microinjection with Delic 12 dye. And as we see here, these cells labeling with fluorescence dye, Delic 12, and we image this with phase contrast, fluorescence and phase contrast images. Another step, it will be zebrafish handling. As you see here, this is the eggs of zebrafish. And here we have embryo. This black dot, it is contaminated eggs. It is not easy to get all eggs in a healthy way. So we have to decorinate these embryo to get out the egg and embed it in gel like this direction. It should be in the same direction, and put it under microscope to injected cells here in this very small area under microscope. Here we have 2 plates under microscope ready for injection. It is so small, as you see here. This is 1, 2, 3, etc. Okay, this embryo embedded in the gel and ready for injection by using microinjection technique under microscope. And here we have embryo 2 days post-fertilization zebrafish embryo after injected with labeled cells with fluorescing dye. Here, this area called privet line cavity. This cell will inject in this area. Because if cells can move to another side of zebrafish, the cells means has ability to move from side to another side. If not, it will stay here in yolk state. Here we have 48 hours post-injection, fish were mounted and imaged again, as described previously using fluorescence microscopy, Nikon microscope. Image montage and analysis of cell migration were designed using Fiji software. You see here, the cells go here in this side, in the yolk sac, but no cells go through the body of zebrafish. That means these cells stay in yolk sac. It hasn't ability to move from other sides. Here we have this image shows the microscope, Nikon microscope. It's a different part connect to this microscope to take this images, of course, with computers. Actually, it was my work in each experiment done in cancer research in the UK. Here we have 2 images about zebrafish. The first image shows that cells can't move and stay in the yolk sac, but the other one, cells disseminate to other part of zebrafish body. And here we have this section from the zebrafish body shows these cells, which already labeled with fluorescing dye. This experiment done with multi-process, as we mentioned, we growth cells, we treat cells. If you want to stop migration of these cells, we stop different signaling pathways by using siRNA and to stop the signaling pathways from the activation and block some sites in the receptors. For example, we have S100A4, S100A6, and inject these cells after treatment in the zebrafish model and image these cells after 1 hour from injection and after 2 days of injections to show the dissemination of cells. Then we collect these cells and do it with a Western blot to see how this protein or gene blocked or active after a treatment. So this result, what we've got from many treatments or many genes to stop the migration of cells. For more details, we can go through this paper, which published in Nature. Here my name, the third authors in this paper, and also for more details, you can use this link to follow this experiment and for more understand more information about this new models. Finally, I would like to thank you for listening. If any question, I'm ready.
Thank you for this lecture, dear Doctor.
Thank you.
If there are any questions, please raise your hand. Thank you. So now I will transfer to my colleague, dear Dr. Haider, to manage the event. Dr. Haider.
Thank you, Dr. Ali, my colleague in the preparatory committee. Thank you to everyone speakers for the valuable information. Now, Chief Chair, Professor Dr. Nauras Abdelilah Alwan, Head of Physiology Department, College of Veterinary Medicine in Basra University, and Assistant Professor Dr. Zahida Alwan, Faculty of Science, Al-Qadish University. Please kindly, Professor Dr. Nauras Abdelilah.
Hello, Dr. Haider. Thank you. Hello for all. With my great appreciation and respect, you're welcome in 10th International Conference of the Arabic Society of the Stem Cell and Molecular Biology. I am chair for this session. I am Prof. Dr. Nauras Abdelilah Alwan with the Assistant Prof. Zahida Marine Hussein. The first oral presentation, Assistant Prof. Dr. Atilaf Al-Mudaffar for the reporter or the research title Cell Therapy and Tissue Engineering, wrote of to the translation of the remote control regenerative technologies for the orthopedic animal model. Assistant Prof. Atiyah.
Hi, everybody. Do you hear me?
For review of the presentation in the 15 minutes. Okay.
Yeah, thank you.
Yes.
And did you see my screen now?
No. Yes.
Okay.
Yes. Okay.
Good morning, everybody. Hi. I'm so happy to be here in this event. Let me introduce myself. I am Assistant Prof. Dr. Itilaf Al-Mudaffar from Department of Surgery and Obstetrics, Veterinary College, and from Baghdad University. Today, I will go through to my work in PhD in Institute for Science and Technology in Medicine in Keele University, UK. I will go through introduction first, and then I will talk about my work. This work is done by Itilaf Al-Mudaffar, Yvonne Rimwald, and Alicia Al-Haj from Keele University Institute for Science and Technology. Firstly, let me define what is regenerative medicine or what it's tissue engineering. Regenerative medicine is replacing or regenerating human or animal cell, tissue, or organ to restore or establish normal function. And tissue engineering is a part of regenerative medicine, which uses the combination of cell therapy, biomaterial, and growth factors to meet the goal of restoration of normal function by regenerating damaged organ. So we have example here, which is a critical size bony defect. And when this organ loses the function, we can restore it by different manner, like autograft, xenograft, or allograft, in addition to tissue engineered way by using autologous stem cell and bony substitute. So because adult bone marrow derived mesenchymal stem cell facilitate the tissue repair and regeneration, the therapeutic approach in musculoskeletal regenerative medicine using adult bone marrow derived mesenchymal cell is promising. Professor Alicia Al-Haj, who is my lead supervisor in this PhD, have developed, and her group have developed a pioneering biomagnetic technology, which called MICA, Magnetic Ion Channel Activation, which is designed to remotely deliver direct mechanical stimuli to individual cell in vitro or in vivo to promote osteogenesis and chondrogenesis by targeting a specific mechanosensitive ion channel on the cell membrane of MSCs and by functionalized biocompatible and magnetic nanoparticle. So this opening of the ion channel can be controlled with a oscillating external magnetic field. And this apparatus, it's computer apparatus established by the MICA Biosystem, which is an American company to produce these kind of bioreactors. This apparatus applied external magnetic field to stimulate the cell in tissue culture in vitro. What are the magnetic nanoparticle? Simply, magnetic nanoparticle is nanoscale magnetic core covered with biocompatible shell. Can use it for with different way to activate the cell or targeting the cell or tracking. And this diagram of the specific canal is called TREC1 ion canal, tweak related potassium channel 1. By this canal, we can track it or tag it with the intracellular nanoparticle, then apply a magnetic field, then we can open or close this ion canal to make the ion charge like potassium here to enter the cell and stimulate the cell to differentiate toward osteogenic or chondrogenic cell.
Sorry, Dr. Tilaf. The presentation is not view and not view for this slide, another slide. Only on the introduction or in the title of the research.
Sorry for that, but I couldn't notice that. I couldn't find the problem. What
close and open another. Close another one or another more.
Okay, let me escape. And,
yes.
Yeah.
No, but okay. Yes. Or come on. Okay.
Now.
Excellent. Yes.
Maybe you press the F5 function.
Come back to first.
May you press the F5 function, please?
Another one?
Yeah. Maybe you press the F5 button.
Okay. So here we present the track 1 canal, which is a mechanical sensitive ion canal in cell membrane. And here is a TRPV canal. And also it's mechanosensitive, which called the transient receptor potential subfamily 5, number 4. And this also we target by the nanoparticle and stimulated by external magnetic field. To select appropriate treatment strategy to achieve a successful or secure healing, animal-based study must be carried out before their successful translation into human practice. Although most studies are focused on orthopedic repair applied on small animal, which is a rodent, better clinical application and repairing orthopedic defect in large animal must be studied. The sheep was most representative animal model for human orthopedics because they are similar in weight, bone structure, and both of biochemical and minerals composition, and also the age and hormonal activity mimic that of a human. In this presentation, we'll introduce the supporting work is for a bigger large animal sheep trial in Keele University aimed to evaluate bone repair and critical sized bone defect and osteoarthritic model utilizing magnetic nanoparticle activate implanted in mesenchymal stem cell. So this is the comparison between using the rodent or sheep in orthopedic surgery or orthopedic regenerative medicine. And
Doctor, the same problem, not change in the slide.
I'm sorry, it's maybe because my internet is not so good. So I try
okay.
Yeah.
Okay.
Now it's okay.
Okay.
Thank you very much. So the rodent is available and cheap, easy to handle management, and have the immune deficiency, but its size limitation in the plant, limited biopsy, and limited clinical translation, and limited long-term study. Sheep, instead of that, was age mimic human aging and similar in large bone structure, similar in biochemical and mineral composition, and similar in weight and dorsile. So, but there is disadvantage, which is there is ethical implication and the expensive cost. Our first experiment was to use the nanoparticle to tag the cell and track it into the defect in the bone, into bone defect in vivo. So we have autologous cell isolated from the bone marrow. Then we expanded and characterized the cell by expression to the marker and tri-lineage differentiation into 3 lineage cell. Then we tag it by label it the cell with nanoparticle, then directly without stimulation the ion canal, directly use it as injectable solution for bone defect treatment in vivo. And here is my my article, which is published in Nature, NBJ Regenerative Medicine. And this is my name, which is 5 in this article. The biggest experiment that I have done in my PhD was the creating tissue engineered cartilage and compared are tested and compared it with a native sheep cartilage. So here we also have 13 donors. We characterize the cell, and also we do a donor variation. If there is a variation between the donors that affect or influence the outcome that we can get it from the stem cell therapy. And we have also characterized the cell and the CD marker suppression and tri-lineage differentiation. Then we use or we choose the best donor and the lowest performance donor to make tissue engineered cartilage by labeling the ovine mesenchymal stem cell with the TREC antibody. Then we applied MICA, which is magnetic ion canal activation, and it drives the cell to differentiate to the chondrocyte. Then we take this engineered cartilage and compare it by mechanical test, histological and immunohistology, and biochemical test. Here we have the cell, and we have the mesenchymal stem cell, which isolated from the donors, and compare it with the chondrocyte to see if the TREC1 and TRBV canal was expressed on the cell, so we can use it by targeting it by magnetic nanoparticle. So there is comparison here between the cell itself and the native sheep cartilage cell, and there is a negative control. Here, one of the comparison after assessment, our tissue engineered cartilage with the native sheep cartilage, and we found that dynamic tissue engineer, which is suffer from applying a magnetic nanoparticle, a magnetic field, and compared with the static that didn't receive any magnetic field stimulation. Here also the comparison between the TRPV4 and TREC1 ion channel, and compare the static with the dynamic part of this stimulation cell. Here we have the biochemical assessment. Firstly is for GAG, which is glucose aminoglycan, the component of the extracellular matrix of the cartilage, and total protein and total collagen, and compare between the both TRBV and TREC1 activation. Here we have histology with 3 kind of dye or staining, hematoxylin and eosine, picroceras, Alcyon blue. Picroceras is for specific stain for collagen. Alcyon blue is a specific stain for gut. Here we can find the comparison between the mechanical stimulating cell inside the hydrogel in vitro, which is the tissue engineered cartilage, compared with the native sheep cartilage histology and acellular, which is negative control. Also here we do the immunohistology for collagen 2, collagen 10, and aggregate, the main content of the cartilage material. And also we compared it with negative control, a cellular gel, and with positive control, which is a native sheath cartilage. In the end, I have to thanks my Iraqi Ministry of Higher Education for support me and for finish my PhD in the UK. And a special thanks to Dr. McLean from University of Nottingham for her assistance and collecting sheep bone marrow and native sheep cartilage. Also, a specific thanks to Professor Alis Al-Haj.
Thank you, Dr. Etilaf. Any question for all, for Dr. Etilaf, for your research?
Thank you very much.
Thank you, Doctor. Another research and the presenter, Dr. Salah Al-Omari, for the title of the research, Rescuing the Functional Defects and the ameliorating, regenerating capacity of the hypertrophy muscles via the genetic manipulation. Please, Dr. Salah.
Okay, can you hear me now?
Yes, thank you.
Okay, is the screen clear for you?
Yes. Yes,
Again?
okay.
Okay,
thank you. Okay.
How long I have? I think 15 minutes, right?
15, yes. Maybe
okay.
20. Maybe
okay.
20.
Okay.
Okay.
Thank you very much. Thank you very much.
Thank you. Yes.
Good morning, everybody. Saleh Omairi from Wasf University College of Medicine. I would like to thank you for imparting us the opportunity to present our data in this conference, the era of biotechnology and personalized medicine, the 10th International Conference of Arab Society of Stem Cell and Molecular Biology. Going to let's remove this screen. Yes, it's okay. To speak about my work that I've done recently in rescuing the functional deficits and ameliorating regeneration capacity of hypertrophy muscle via genetic manipulation. In this 15 minutes, I'll try to give like a brief introduction and background about my topic, the main molecules in my study. I would also go quickly about the hypothesis and the aims of our experiments. What are the main methods that we used in this experiment? And I will refer to the main results at the same time with a quick conclusion and a quick reading for the future directions and the main publication that I've published in relative to my work. So first of all, skeletal muscle is a highly complex tissue that undergoes both qualitative and quantitative change as a part of its adaptation to the physiological and environmental stimuli. A central notion or central concept of skeletal muscle biology is the trade-off or the existence of an inverse relationship between oxidative capacity and muscle fiber size, which might explain why slow muscle are small and fast muscle fiber are large. So the small muscle fibers are highly oxidative muscle fibers characterized by high number of satellite cells and high cross-sectional area with high content of mitochondria and myoglobin and denser microvascular network than the fast muscle fibers that are predominantly depending on glycogen for the ATP production. Thus, they are glycolytic fibers. Importantly, skeletal muscle, this important tissue, can change in terms of not only the mass, in terms of fiber number and size and composition, but also the supporting tissues could be changed with the muscle fiber change, such as the capillary density, the blood supply, and the connective tissue. I would give an idea about one of important molecule in my study, which is myostatin. Myostatin is a potent inhibitor of skeletal muscle development. It is a negative regulator of the muscle growing. And when we delete myostatin via the knockout of this gene, there is a muscle doubling phenomena could be seen in mice, cattle, and even in human. In addition, there is a deficit in mitochondrial and blood supply efficacy of these muscles when we delete or knock out myostatin. In addition, we found that there is a reduction in number of satellite cells. Maybe this image gives you an idea. So when we compare this myostatin knockout, when we delete it with the normal, with wild type, as well as in the dog, you see how muscular they are. Another important molecule is estrogen related receptor gamma, which is a master regulator of skeletal muscle metabolism and oxidative capacity. It is an orphan nuclear receptor that has been used to induce the oxidative phosphorylation in muscle tissue. It has been shown that the number of satellite cells increase in muscle when they become progressively oxidative. So the hypothesis of our work, we trying to develop large oxidative fibers without compromising the functions such as the fatigue resistance, the oxidative phosphorylation, and the regeneration capacity. In order to do that, we developed a novel mouse line in our lab by introducing of ERR gamma, estrogen-related receptor gamma, as I mentioned previously, overexpression into the myostatin null background muscles. So just to make it easy for you and for me, we have three generations in our study. The wild type, which is the control. The second one is the myostatin null, where we are deleted the myostatin gene completely by knockout process. And the third one, when we overexpress ERR gamma, only the myostatin null background muscle. So I will call the ERR gamma. So we'll have wild type, myostatin null, and ERR gamma, just to make it easy for you when I explain my results. I would also just give an idea about the degeneration regeneration cycle. When our muscle damaged, directly the degeneration started, the accumulation of the necrosis muscle fibers and necrosis tissue. After that, within about 24 hours to 48 hours, there is an inflammatory response indicated by the presence of neutrophil that produce chemical substances, give us signals for the monocyte to come to the site of damage. Monocyte then stimulate macrophages, convert to the macrophage type 1. From day 1 to day 3, after muscle damage or trauma, the macrophage 1 will be conveyed to macrophage type 2. So the macrophage type 1 will be responsible to remove all the debris as a result of muscle fiber damage, stimulate pro-inflammatory cytokines. When they convey to macrophage type 2, the anti-inflammatory cytokines production will be start and then stimulate satellite cells. When satellite cells, which are the stem cell in the muscle, and the skeletal muscle, stimulated by macrophage type 2, the regeneration started. And the stem cell will be migrate toward the site of the damage to start the regeneration process. After that, there is an after the regeneration, after the forming of a newly formed embryonic myosin heavy chain, there is an repairing to the tissues that connect to the muscle, such as the connective tissue in the extracellular matrix, and angiogenesis, I mean the blood vessels. And within about 15 to 20 days after the damage of muscle degeneration, we will have the full maturation indicated by even the neuromuscular junction repair. So I would like just here to explain main methods that we use in this part of my work. So we worked on the hind limb muscles, such as the tibialis anterior muscle, EDL, which is extensor digitorum longus muscle, gastrocnemius muscles. What we have done in these muscles, for the tibialis anterior TA muscles, we investigate the ratio between muscle fiber and the blood vessels by using the CD31 as a marker, as well as we investigate the number of necrotic fibers, the macrophage density indicated by the F480 marker, as well as the apoptotic cells to determine the number of death cells in all three genotypes, wild type, myostatin null, and ERR gamma. And here in the apoptotic cell, we use the Cleveland-Caspase-3, and we try to determine which muscles are produced and during the regeneration give us a new embryonic myosin heavy chains. And we use also finally the PAC7 and MyoD to indicate the committed cells. Gastrocnemius muscles have been used for the molecular work because we can get large muscles and so large tissue and result more RNA. Succinate dehydrogenase has been applied to determine the oxidative capacity, and we also use the EDL for the immunocytochemistry for the single fiber analysis. So moving to main results of our study. From the first bit, we notice that there is an increase in muscle mass following myostatin deletion. As you see here in pictures, we can see that in the picture in the middle, there is an increasing in the muscle mass compared to the wild type. So we weight EDL muscle, extensor digitorum longus muscle, gastrocnemius muscle, soleus muscle, and tibialis anterior. We found that there was a significant increase in muscle mass. All the tested examined muscles, we found there is an increase in muscle mass in the myostatin null mice compared to the wild type. Importantly, muscles from ERR gamma that we overexpressed on myostatin null background maintained the increase in muscle mass exhibited by the myostatin null, as you see here in the pictures, but with different color. Then we tried to know whether this increase in muscle mass resulted due to an increase in muscle fiber number or muscle fiber size. So we measured the cross-sectional area of all muscle fiber type in muscles, and two muscles, EDL and soleus muscle, because EDL is a mixed muscle and soleus is an oxidative muscle. We found that all muscle fiber type show an increase in the size, indicating by large cross-sectional area from myostatin null compared to wild type. Again, interestingly, the same size we found in the ERR gamma. Then we moved to count the number of muscle fiber. Again, there is a significant increase in number of muscle fibers in muscle from myostatin compared to the control, and ERR gamma maintained this increase in muscle fiber number. So we can say the increase in muscle mass that we just noticed is a result of both hypertrophy and hyperplasia. Then we moved to indicate the, metabolism status, and we have applied the succinate dehydrogenase protocol for all muscles of our study, EDL, soleus, and tibialis anterior in the deep and superficial areas. We found that there was a significant reduction in number of oxidative muscle fibers in the muscle fiber of myostatin null. As you see in the red color, there is a reduction. However, when we express and do superimposition of ERR gamma, there is a restoration of number of oxidative muscle fibers to that level of wild type, which is the control. And we so the same scenario in all examined muscle, EDL, tibialis anterior, deep and superficial, and the soleus muscle. Then we investigate the angiogenesis program at protein and transcriptional level. So we investigate the expression level of main genes that are controlling the angiogenesis program, which are vesicular endothelial growth factor alpha 165, vesicular endothelial growth factor alpha 189, and fibroblast growth factor 1. We noticed clearly that there is a significant decrease in the expression of these genes in muscles of myostatin null. However, overexpression of ERR gamma show that there is an elevation in the or upregulating of these genes, not only more the myostatin null, but even significantly higher than the control. That was at transcriptional level, as I mentioned. Then we counted the number of blood vessels in each muscle fiber by using CD31 as a marker, and we found that there is a reduction in number of blood vessels by each muscle fiber from myostatin null, as you see in this middle picture. And after introducing of ERR gamma, we found that there is a restoration and there is an increase in number of blood vessels per each muscle fiber. Moving forward, at a single muscle fiber, we tried to count the number of satellite cells. Why? Because satellite cell is the main cells that muscle depend on it for the regeneration process. We found, and we used BAC7 as a marker for the quiescent satellite cells, MyoD for the proliferative and activated satellite cells. As you see, there is a just single fibers. We separate the muscle fiber of EDL. Just to mention, actually, I have done this separation of this muscle fiber by my hand, and I got about 800 to 1,200 single fiber from each muscle. It was really hard work. So what we found that there was a significant reduction in number of satellite cells per each muscle fiber, and in total, in the muscles from myostatin null compared to wild type. Again, we found in using that the number of satellite cells and ERR gamma even lower than the myostatin null. So, and that was in T0 and T48. So at time of 0 and then after 48 hours. So from the previous results, everything was good by ERR gamma overexpression. We maintained the size, we introduced the angiogenesis and the blood vessels, we introduced the oxidative capacity. However, there was only a bad thing, or let's say unexpected results. There was a reduction in number of satellite cells. So in order to investigate whether the reduction in the number of satellite cells would affect the regeneration capacity of these muscles or no, so we moved to the second experiment. We introduced the muscle degeneration of the mice from 3 genotypes, from wild type, myostatin null, and ERR gamma. We used cardiotoxin, which is a venom from the cobra snake. We inject it to the tibialis anterior. Then we investigate the regeneration capacity at 2 time points, which are day 3 and day 6. Why we select these 2 time point, because with day 3 we expected that everything has been done in terms of debris removing. Day 6, when we're expecting to see a newly muscle fiber. Let's see the results. So by day 3 of after degeneration, after the injection of the cardiotoxin, we found that there was a high number of necrotic fibers in muscles from myostatin null, and the number of necrotic fibers has significantly reduced and decreased in the muscles from ERR gamma. As I mentioned, we have 3 genotypes: wild type, myostatin null, and ERR gamma. Then we looked to the density of macrophages using the F480 as a marker for the macrophage, and we found that the density of macrophage was significantly higher in muscles from ERR gamma compared to the myostatin null and wild type. By day 3, also investigated the number of the committed cells using BAC7 and MyoD on the tissue, and we found that there is high number in the ERR gamma of the committed tissue, I mean the of the committed cells, I mean the cells with MyoD positive, which are active and ready for proliferation and differentiation to go to the next step. By day 6, we repeat 3 parameters that we have done in day 3 to investigate, and we confirmed our result. Still high number of necrotic fibers, I mean the second row of the images, if you with me, and there was also a reduction in number of necrotic fiber in the muscles from ERR gamma, as you see here, and in the IIGG. Then we moved to look to the macrophage. Still high number of macrophages in the ERR gamma compared to other 2 genotypes, and more of the committed satellite cells that express MyoD in the ERR gamma compared to the other genotypes. What we have done extra in day 3, we looked whether the number of apoptotic cells is the same or different between the 3 genotypes by using a clipped caspase 3 in our tissue for the immunostaining. And we found that the number of apoptotic cells, the dead cells in the ERR gamma, significantly lower than the number of the dead and apoptotic cells in the myostatin null and wild type. Which interpretation for that? Maybe all the muscles or all the muscle fibers has been utilized, or all the cells have been utilized for the newly formed of embryonic myosin heavy chains, which indicated here as the first row of the image. And we found that there was high number of the embryonic myosin heavy chains, which are the newly formed embryonic myosin heavy chains in the ERR gamma compared to the myostatin and wild type. So, in conclusion, muscle specific expression of ERR gamma in the myostatin null mice maintained the increase in muscle mass resulted from myostatin deletion, which is good. Oxidative program imparted through superimposition of ERR gamma in myostatin null induces proposed angiogenesis program, which is good again. Even though the muscles of ERR gamma have fewer satellite cells, their muscle regenerating capacity exceeds that of both myostatin null and wild type. So even with low population of satellite cell, there is a good regeneration capacity. So in summary, our work challenges the dogma of that there is a trade-off or an inverse relationship, as I mentioned in the beginning of my speech. We challenge the dogma that there is a trade-off between muscle fiber size and oxidative capacity, and indicates how we challenge the dogma, because we produce large oxidative muscle fiber. And the second part, we indicate the important role of microcirculation in the regenerative capacity, even when satellite cell number are reduced. So In the future, we would like to determine how connective tissue adapts with the change in the muscle fiber. And we also would like to investigate to what extent that ERR gamma can attenuate muscle function in starved myostatin if we put them in the starvation diet for whatever time. We also looking to investigate if similar phenotypes could be obtained via non-genetic modification. I mean, the post-developmental of myostatin blocking, is that possible or no? And finally, we will try to determine the ability of ERR gamma to confer resistance to the obesity in response to concomitant high-fat diet. So thank you very much for your listening. And sorry if I take more than my time. I would just to show you just some of my publications recently in the eLife Sciences, the first one in the left corner, and which is with impact factor 9.3. It's UK, and the molecular therapy nucleic acid, 3 of them in the scientific report from the Nature publisher, and the last 3 was from 2 of them in the neuromuscular disorders, and last one in the cachexia muscle journal. Again, thank you very much for your listening. If you have any question, I'm ready to hear from you.
Thank you, Dr. Salah, for this excellent presentation.